Morning. So welcome back for another session. So there's a slight change of plans. Maithreyi will speak for half an hour, and I've been nearly arm twisted to talk about the mechanics part of the thing in this session. So she'll talk for half an hour, and then I'll have one hour session on the mechanics aspect that we didn't cover yesterday.
Okay, so what I'm going to try and do today is go from looking at how you sculpt tissues that I described yesterday were all monolayers and relatively simple, to how you build an organ. And the organ I will choose for today's lecture is the eye. And much of what we think of when we see the eye is something that looks like this. But the development of the eye is actually a very complex morphogenetic process. And as you will see in this cartoon, kind of like a cross section, the eye actually begins as an outpouching from a part of the brain that's called the diencephalon. So it forms a little outpouching like this that we will call the optic vesicle. And you can see that the shape of this optic vesicle changes from being this sort of U-shaped structure, let's say, to becoming curved again, so that from having just a single outpouching, you've created a structure that looks like this. It eventually has a part of it oriented like this, the other part of it oriented like this. So what you will also see is that this outpouching is makes contact with a monolayer of cells that's called the surface ectoderm. And studies in mouse models have told us that the interaction between this outpouching and this blue surface ectoderm are actually necessary to drive the further morphogenetic changes that accompany the development of the eye, right? And you'll see two things happen. Not only is the green structure getting deformed, the surface ectoderm that's above it is also getting deformed, right? So in some simple way, you can think of what's happening in the surface ectoderm along the lines that I described to you yesterday, which is like what happens during dorsal closure, what happens during neural tube closure, meaning that you have a sheet of cells and you have something that induces that sheet of cells to bend and create this sort of invagination. But what's happening in the green structure is a little bit different in that it's now forming a more complexly folded structure, so that this actually becomes a two-layered cup at the end of it, right? And this flat layer first forms an invagination, and then that blue bit in the middle actually detaches from the surface ectoderm, become this structure, which is the lens, and then the surface ectoderm covers it. So in simplistic terms, you might say that what's happening in the blue part is similar to what I described as happening during ventral furrow formation, where you have apical constriction of these cells that leads to bending or invagination of that sheet, then internalizes to form a tube-like structure. Except that although the neural tube stays as a tube, we saw that the ventral furrow or the mesoderm actually then flattens out and spreads along here. What's interesting is also what happens in this green structure, which is that it ends up forming a double-layered cup. And if you ask why this is important, the two layers of the cup actually end up forming two distinct structures. The inner layer of this cup forms the retina, which actually allows us to see or sense light. The layer on the outside forms the retinal pigment epithelium, right? So this is actually quite a complex structure. And one of the reasons I decided to pick this structure is twofold. One, because it's a structure in which it's been demonstrated by studies in animal models that this process requires interactions with neighboring tissues, right? So if you just look, so this is now turned in the opposite direction, but what you can see is that as this green structure is forming, this is the surface ectoderm, that blue layer here. This is the green bit here. And you can see something here in blue, right? So essentially what studies in animals have shown is that this surface ectoderm, this green structure, as well as the tissue around it, which is a mesenchymal structure, all are sources of signals and interactions between the green and blue and the gray and blue are necessary to acquire this final morphology, this optic vesicle, but to also acquire the morphology that allows you to form this lens in the middle. So all I'm using this to highlight is that there are sources of signals like morphogens coming from here, morphogens coming from here, morphogens coming from here. Some are the same kind, some are different kinds. The arrows indicate some sort of hierarchy in gene interactions, and these double arrows indicate interactions between tissues that are necessary to pattern this entire structure. What I want to focus on is not the details of these gene regulatory networks and signals that pattern this structure, but to simply ask, do you actually need these inductive signals from all these surrounding tissues to get this complex morphology, or can you do away with it? And so that's really the question I want to ask here. So how much of complex stereotypical morphologies that we see in living systems actually require induction, or the complex spatial distribution of signals both within the tissue and outside it, and how much might actually happen in the absence of such complex patterns. And the reason I want to ask this question is because it allows me to talk about morphogenesis in a dish. And I would say one of the best examples to date of being able to create a complex structure in a dish is the making of an eye in a dish. So let's not get into whether this eye can see. That's not the purpose of this talk, but just to see what the level of complexity of pattern formation you can get in a dish is in the absence of all these surrounding tissues and influences that normally accompany development. And the only chemical detail that I would like you to remember here is that a transcription factor, which we've already discussed, is a protein that binds DNA and directs transcription of target genes. A transcription factor called Rx or RAX directs this entire morphogenetic process. And in animals that don't have this transcription factor, you have anophthalmia, which means you don't make an eye. So just to spend a little time to talk about induction because I've not used that word in my lectures so far. So embryonic induction is essentially a term that's used to define the ability of, let's say, a group of cells or a tissue to influence the state of differentiation, for example, of another tissue to which it's near which it's placed. And the really striking experiments that established embryonic induction were the experiments of Hilda Mangold and Hans Spemann, who showed that if you actually cut out a piece of tissue, a particular piece of tissue from a frog embryo, and transplanted it into a different location in a host frog embryo, that bit that you transplanted would actually give rise to structures like the vertebrae or the somites and notochord and so on. But it was also capable of inducing the host structures around it to become the neural tube. And subsequently, a whole system of organs developed around where the graft was placed and grew into a nearly complete secondary embryo, generating these sort of two-headed newts. This suggested that there was something about this pink region that was transplanted that had the intrinsic capability to give rise to some kinds of structures by itself, but also the capability of changing the properties of the host cells around it, and thus having a huge influence on the tissue near which it was placed. And this was the fact that it could induce the neighboring tissue was evident from the fact that the second axis or embryo that developed was composed partly of grafted tissue and partly of induced host tissue. So that's the spirit of induction, and subsequently people worked out the molecular details of how this region of the frog embryo might function as an inducer. And all I'd like to say here is that at least one way in which it can do it is to act to inhibit a particular signal that's otherwise uniformly present within the frog embryo. And it can now induce neural structures like it did in the previous slide by inhibiting that signaling pathway. So it's a source of inhibitors for a particular, the ligand of a particular signaling pathway. Let's not get into what the details are, but that is how at least one of the molecular attributes that allows it to function as an organizer. So I want now to turn to induction versus self-organized morphogenesis. I said to you that studies in animals have shown the importance of tissues surrounding the optic vesicle in driving the morphogenesis of the eye. But what Yoshiki Sasai sort of bravely asked was whether you could actually do away with a lot of these inductive influences and still generate fairly stereotypical but complex shapes. And if so, what mechanisms might contribute to this? And so there are reasons, I mean, there is evidence to suggest that cells or cell collectives can actually generate quite complex patterns in the absence of very complexly patterned signals in their environment. One of these is the ability of cells to sort themselves out, cells in mixtures to sort themselves out. I'll spend just a minute on this to say that it had already been shown before Sasai ventured into his experiments that if you took took cells that were derived from different germ layers of the frog embryo, namely ectoderm, mesoderm, endoderm, then dissociated them so that they were now single cells, and mixed them back together so you had this salt and pepper mixture of, so to use a word that people here have been using, well-shaken mixture of cells of from different germ layers, and let them aggregate and stay for a while. What was observed was that the cells will actually sort out. The green cells will sort out from the red cells. One cell population will typically envelop the other population. And if you do this with combinations of cells, the same population would always be found on the outside and the other population would be found on the inside. And so Steinberg and Holtfreiter, who performed these experiments, suggested that this ability of cells to unmix or sort out from each other must rely on some differential affinity that these cells have for each other. And he said that, so I'll just come to what he postulated was responsible for this, but he proposed that the forces organizing the cells are closely analogous to those organizing the molecules of immiscible fluids. And he suggested that maybe varying degrees of surface adhesion between cells causes them to spontaneously reorganize for reasons to minimize surface tension, that sort of thing. So this differential adhesion hypothesis that he postulated, he suggested must arise from differences in surface tension that in turn must arise from differences in the adhesiveness of these cells. And the explanation for this, as I already mentioned, is to meet the tendency of cell clusters to minimize their surface, similar to how you minimize surface tension in liquids, right? So that sort of thing had been observed. So using, given that backdrop, what Sasai sort of set out to do was to ask if you could achieve these sort of complex behaviors in a dish. And he started out with an aggregate of homogeneous cells, embryonic stem cells that we discussed in the first class, but embryonic stem cell aggregates that had been induced to be capable of becoming that green tissue that I showed you, that's involved in the entire morphogenesis of this vesicle. So if you induce these homogeneous cells to become that retinal neuroepithelium, using that same transcription factor that I spoke to you about, what they found was that this aggregate, after this transcription factor became expressed, showed that in the cells in which this transcription factor was expressed, evaginated, meaning put this out pouch, just like it happens normally during optic vesicle morphogenesis. But it didn't stop at that. This evagination progressed to form the invagination that results in the formation of that two-layered cup that I showed you, right? What they also found was that if you look at what the inner and outer walls of this two-layered cup are, just like in the normal eye, they showed that this inner layer is molecularly like the neuroepithelium, or the neuronal retina that allows you to sense light. And the outer layer is the retinal pigment epithelium. What they also showed was that this cup is formed with the apical end of the cell oriented in a convex kind of way, which is different from here, where the apical end that's constricting is concave. So essentially what they showed was that embryonic stem cell derived retinal epithelium spontaneously formed hemispherical vesicles that became patterned along this axis. And this occurred in the absence of a lens or surface ectodermal tissues that I showed you, provided inductive signals during the normal development of the eye. And so this is essentially what you see as these aggregates develop over time. You will see the green bit evaginates first, and then it forms this two-layered cup. And you can look at all the molecular signatures of what these cells have become. And this is how you know that the architecture of these of this two-layered cup is like the architecture of the two-layered cup from a real embryo. So this just quickly shows you a movie of this. So you will see over time how these vesicles form. Basically, you can see that just from an aggregate and one transcription factor, you can get this complex morphology. This just then shows you, I think, how the double-layered cup actually forms. And then there's one more that shows you, I think, well, so there are other examples. So yeah, you can see that two-layered cup happening. So basically, very stereotypically happens in culture. And then what they went on to show was that this subsequent differentiation of the neuronal epithelium and the retinal pigment epithelium required the prior differentiation of the neuronal epithelium. And then if you took it away, you did not differentiate the second layer into the pigment epithelium. So in this dish, what you saw was that temporally this green structure first differentiated into the neural retina and then induced the cells around it to become pigment epithelium. So you can achieve induction of the two cell types in addition to morphology in this dish. And this just tells you that you require that progression of this one, of the neuronal retina forming first to induce other to happen. And if you look at the architecture of this neuronal retina, it's like what you would see in the real eye, right? So I'm not going to describe the cell biology here. But so it has very closely resembling features to development in the real eye. And so then they also went on to find that you require both cell division and actomyosin contractility to generate this cup-shaped morphology. Surprisingly, what they showed was that you required actomyosin contractility only in this stage. But if you messed up with actomyosin contractility at this stage, nothing really happened. Likewise, if you messed up with cell division, there was a change. You didn't nicely form this two-layered cup. So what they argued was that somehow these aggregates are also spatio-temporally regulating their actomyosin contractility and cell division to enable this morphology. And okay. But surprisingly, what they found was that if you looked, so I told you that morphologically the structure looks like what happens during ventral furrow formation. But what they found was that if you look at this two-layered cup, all of the phosphorylated myosin that indicates active contractility is located here, but there's nothing in this part that's actually formed this cup, right? If this cup were constricting apically, and that apical constriction were responsible for the invagination, you would have expected this to be apical and this to be the region where you accumulate active myosin. But in fact, there's no active myosin here at all. All of the myosin that you see is in the pigment epithelium layers. And you look at this, therefore, the apical end is here, the basal end is here. So this is apically convex rather than apically concave, as we saw in the ventral furrow invagination. And for you one more movie to. So they use laser ablation as a way of figuring out whether there's anisotropic tension. And so they use multi-photon lasers to create little ablations and saw what the tissue response was. Basically, the two ends of the tissue actually came back close to each other. What they found was that this response was disrupted. So they did it in different regions. When they did it in the hinge, for example, as you will see just now, this did not happen. They didn't come close to each other. And when they disrupted cell division, also these recoiled edges didn't come back close to each other. So this allowed them to map the two walls or different regions in the walls of the cup for their mechanical properties, and suggested that that a combination of locally regulated contractility and cell division must contribute to this morphogenesis that's happening in a dish. So I'll just tell you what they inferred from experiments of this kind. They said that the invagination of this retina occurs without forces or signals from external structures, and therefore it must be driven by a sequential combination of local rules and internal forces within the epithelium. The second was that even without the surrounding tissues, you get the patterned differentiation of this vesicle into the neuronal retina and the retinal pigment epithelium. And that in the distal neuronal retina, you can see a reduction in stiffness. I didn't show that to you. I'll show it to you in the next slide. And that this could be because there's no actomyosin accumulation in this tissue. So in a way, you could ask what drives the apically convex invagination here. And they argued that it could be one of two things. One is that at the boundary or the point at which you start to form the two layers, by that I mean the hinge region, you have apical constriction, and this causes the retina to become passively invaginated, and or that the proliferation that happens in the retina facilitates the invagination. And what I just want to end with showing you is actually something that you will probably hear a lot of in the coming talks, which is that using a combination of experiments and simulations, including the use of 3D vertex models, and putting in parameters derived from experimental measurements, what they showed was that this bending of the neural retina so that it's now apically convex. So both these features, the bending of the neural retina and the formation of that hinge region, were captured by their simulations. And the simulations suggested the dependence on how much of each tissue you had and on cell proliferation. But really that a combination of experiments and simulation actually showed that that reduction of actomyosin accumulation in the neural retina on the apical surface is what bends the epithelial sheet in the apically convex direction. So this allowed them to argue that in a dish, in the absence of all these external signals, the neural retina generates autonomous bending force in the apically convex direction, and that actomyosin reduction relaxes apical contractility, and this contributes to its bending in the convex direction. What they also showed that hadn't been seen before, and they saw it for the first time, was that the cells at the hinge actually shorten, right? This is not apical constriction, but they become short, and that this constriction actually also contributes to this complex morphology here. So I will end with this last slide to tell you all of what they've been able to do in a dish. What they've been able to do in a dish is cause autonomous differentiation of this optic vesicle into the two layers that make up the retina. They've sort of uncovered how you can bend this retina in an apically convex direction. And what they've shown by using mechanical perturbations of the kind that I described in other experiments, either pushing a micropipette on this spherical or hemispherical stage and so on, was that this bending force actually propagates to this hinge region here and then induces lateral constriction of those cells, rather than what people had predicted before, which is that apical constriction in the hinge region passively invaginates the neural retina. And so one of the things they're suggesting is that the way you can generate a complex architecture such as this double-walled cup in a dish in the absence of external signals is by, so essentially what they've demonstrated is once you have cells that are competent to become retinal epithelium, almost everything else follows from there without having to add any external signals. They follow in a spatio-temporally evolving mechanism. So essentially, what I think this sort of work shows is that you can have deformation-triggered mechanical feedback and self-organization that together contribute to complex but stereotypic tissue shapes. And what this requires is intricate local control of forces and tissue material properties. And so this, I think, allows you to think of how you generate complex patterns outside of an animal and then begin to wonder what is actually necessary, what happens just because of what is available around it and what can happen in their absence. So I think this is a nice demonstration of what can happen in the absence of a lot of complex cues from the environment. So I will end there and let Vijay continue.
I'll try to give a brief picture of how one thinks about mechanochemical patterns. And so to get to that, we go back and think about kind of things that we've been writing and how to connect them back to what we learned about stochastic process. So if you remember, we had the master equation for any arbitrary stochastic process with some discrete set of states. So I'll just write that down. You had gain term and a loss term. So you ask the following question. If I have a stochastic system, for example, some set of molecules in a container, and they have some discrete states or even a continuous set of states, and the dynamics is such that there is some master equation like description. We've seen several examples, chemical reactions, motility of molecules, and so on. When is it that you get a steady state? So a steady state would in general mean that dp by dt is 0 for all states x. So if this goes to 0, that would be a steady state. So that would actually imply that this entire sum would go to 0. Right? It doesn't tell us anything about individually the transition rates W's or the state probabilities. All it tells us is that the sum of all these terms should be equal to 0. That's all we need to get from that. However, if the system is in a particular state called equilibrium, thermal equilibrium, so it satisfies the following conditions of what is called detailed balance. So the condition of detailed balance says that not only the entire sum vanishes, but each term in the sum vanishes identically. So which means that if you look at the probability of being in the state x prime and making a jump to the state x prime, to the state x, it's identically equal to the probability of making a reverse transition. Right? So you have states x and x prime. You can make a jump from here to here, or you can make a jump from here to here. And detailed balance says that these two fluxes are equal. And if they are equal, you are at thermal equilibrium. You are at equilibrium. Thermal equilibrium, this plus specifying the thermodynamic equilibrium, also specifies that the probability of each of these states is directly proportional to energy of those states. So the condition of detailed balance plus the condition of thermodynamic equilibrium or the Boltzmann probability distribution completely specifies a system at thermal equilibrium. You need both. So one tells you how should the dynamics go around, the other tells you what is the state probability. This is important because this is really saying if I have a bunch of states, x, x prime, x double prime, and so on and so forth, detailed balance condition tells you that there are no currents possible between any two states. So each state can make jumps between the others and so on. But if you look at every possible condition, the detailed balance condition says the probability of jumping from here to here is the same as the probability of jumping from there to there, and so for every pair of states, which means identically currents vanish between any two given states. So thermal equilibrium also implies no currents. Okay, so that's the connection to the microscopic master equation picture that we have. But we learn when we do thermodynamics that a system is in thermal equilibrium if different parts of the system are in equilibrium with respect to these three quantities. So you say if you take two parts of the system, if the two parts of the system have the same temperature, then it's in thermal equilibrium. So the thing that tells you whether two parts of the same system are at the same temperature is this quantity called temperature, thermal equilibrium.
There's a part that tells you whether the system is in mechanical equilibrium. That is, this part does not put any net force on the other part and this part does not put. So that means there is a mechanical equilibrium, which means a balance of pressures. There's a thermal or there's a chemical equilibrium, which says the amount of molecules on this side and the amount of molecules on this side must be balanced. Otherwise, there'll be a flux of materials, there'll be a chemical reaction pushing. So there is a chemical equilibrium. And the quantity that tries to quantify that is the chemical potential. So thermal equilibrium, mechanical equilibrium, chemical equilibrium, if all three satisfied, we call it a thermodynamically equilibrium state. Okay? So that's a boring state, like a glass of water left to itself. So with this set of a definition of the thermodynamic equilibrium, we all learn that there are a couple of laws that come up. Basically, energy is conserved, entropy increases. That's all is relevant for a living system. Examples from the living world. If you have a microtubule which is growing and pushing against a barrier, at each point of time, you have a mechanical force balance. The force exerted by the membrane, there's a force exerted by the microtubule, and that's why you have a balance. And that's the calculation that we did. Or you have a bunch of molecules which have some different weights and they settle down under gravity, therefore there's a partitioning. So there is gravity which is doing the mechanical equilibrium part. There could be chemical equilibrium between the unfolded, folded states of some protein, or protonation, deprotonation of some chemicals, ligand binding, unbinding, ion channels opening, closing. There can be chemical equilibrium. These are all examples of discrete states. And so if the discrete states satisfy these thermal equilibrium detail balance conditions, there's no net current. And the probability of opening or closed can actually be calculated by the Boltzmann probability distribution. So that's our usual thermodynamic, equilibrium thermodynamics. However, living systems are not at thermal equilibrium, not at thermodynamic equilibrium. So therefore, how does one describe systems which are away from thermodynamic equilibrium? So basically, you would like to ask the following. If I have a glass of water, I leave it there for some time, it will eventually reach thermodynamic equilibrium. Temperatures balance out, pressures balance out, chemical potentials balance out. But now I'm going to heat it from behind, let's say. So you're supplying a flux of thermal energy, and so that can set up some flows in the system that can push, pull molecules around and so on. So how do I describe this dynamics? How do I describe a non-equilibrium state for the system? So one could, in principle, say, catch hold of every molecule that's there in the glass of water and write down Newton's laws. In principle, yes, one can do that. But in practice, how many molecules are there? 10 to the 23 molecules. There's no one who can solve all those equations, tell you the entire dynamics. So one makes a coarse-grained description. Just like in the thermodynamic description, one did not look at each and every molecule sitting here, but made some larger coarse-grained variables called temperature, pressure, chemical potential. These are not properties of individual molecules. These are properties of aggregates of molecules. So one asks that if a thermal, mechanical, and chemical equilibrium, and so thermodynamic equilibrium, there was uniform temperature, there was uniform pressure, there was uniform chemical potential. If I perturb this uniformity in space and time, I will generically generate a non-equilibrium state of the system. So the system will then try to slowly relax. So a glass of water, you heat it from behind, and if you heat it for some time and let it go, initially it's out of equilibrium. The bottom part is hotter than the top part. Eventually the whole system will relax back and attain a new temperature. So we would like to really extend hydrodynamics, we would really like to extend thermodynamics, and the extension of thermodynamics to the systems, to cases where the thermodynamic variables like pressure, temperature, chemical potential are functions of both position and time is really hydrodynamics. And you really cannot have arbitrary functions of space and time. The functions of space and time must be very, very slowly varying compared to the microscopic scales. So it's essentially you need to have this coarse-grained picture still at the back of your mind. You cannot have variations in the pressure as a function of space on length scales comparable to the mean free path. It doesn't make sense. At that description, the thermodynamic description doesn't work. You really need to have variations much slowly, much longer time scale. Therefore, what one really gets is an idea of a local thermodynamic equilibrium. So you take a patch of the material. Locally, the temperature, pressure, chemical potential are uniform here, but it's slightly different from the neighboring and slightly different from the neighboring thing. So on a very long length and time scales, you have a slow variation of the chemical potential, thermodynamic, temperature, pressure, and so on. So if that is the case, we would really like to understand the following. So you go back to this picture, and let's say the system has relaxed to some thermal equilibrium. and now let's say I actually increase the temperature of the system. So what happens? There will be a flux of thermal energy from this side to that, such that eventually the two systems have the same temperature. If I take this system and increase the pressure here, that will put in a force on the semi-permeable membrane and move it such that the pressures balance. In other words, if you perturb the system, you will get additional thermodynamic forces which will try to restore the thermodynamic equilibrium. Okay? So the perturbations that you would put in terms of pressure, temperature, chemical potential will generate thermodynamic forces which will balance out all the fluxes. So if you leave the system to itself, you make a perturbation, leave it, don't do any more perturbations, eventually it will relax back to thermal equilibrium. So that's a case of systems or non-equilibrium process where it's a process where there is a relaxation to thermal equilibrium at the eventual time scale. Living systems, and particularly non-equilibrium steady states, are systems where you never allow it to reach thermodynamic equilibrium. You constantly flux it with energy. Always ATP being consumed, you always eat something, that's some chemical cycles. At the end of the day, you have a store of energy. And the store of energy is constantly used to do work. So if you don't eat for 10 days, you will reach thermodynamic equilibrium. Or maybe longer, depending on your. So therefore, you keep consuming energy, and so therefore keep driving the system out of equilibrium. So how do we have a description of systems which are driven out of thermodynamic equilibrium? So that's what we really want to do. In general, we don't have a description. If you give me an arbitrary way of looking at a driven system, driven out of equilibrium, and tell me how do I do the hydrodynamics, we don't have a way, we don't have a physical theory for that. What we have is a description for large-scale, long-time properties of systems close to thermodynamic equilibrium. Arbitrarily far from thermodynamic equilibrium, we do not have an example. Weather. We can't predict tornadoes, we can only see them, even though we know the equations that the weather patterns satisfy. So it's a very, very complex system. Now the great unsolved problems of 19th century, turbulence. Okay. So this is what is called a hydrodynamic theory. Hydrodynamics doesn't mean water. It started from water because the theory for water was one of the first hydrodynamic theories. But one basically thinks about writing down descriptions for a many-body system, collective descriptions for a many-body system, on length and time scales much, much larger compared to the microscopic length scales, microscopic time scales. So what one does is to take a many-body system, for example, a glass of water or a pneumatic liquid, and you disturb it away from equilibrium. So you maybe slosh it, heat it, push it, pull it, do something, do some perturbation, and leave it. You will find that the system will relax to itself, relax to thermodynamic equilibrium. Many degrees of freedom will relax very instantaneously, almost instantaneously. But there will be a few collective degrees of freedom, things like momentum density, mass densities, or orientational fields near order phase transition. That will take a long time to relax. So I'll qualify that, what do you mean by long and short? So hydrodynamic theories describe these long-lived degrees of freedom or the hydrodynamic modes. So the relaxation time of these modes, this is the qualification, that if I have a system, let's say a density field as a function of x, and you reach some thermodynamic state, and so which means the density has become some uniform value. Now I take this system and I perturb it. So let's say I perturb it with some wavelength lambda. So I make the density higher here and lower there. Remember, I'm not creating new molecules or destroying molecules. I'm just redistributing the molecules. So therefore, I've created a higher density and a lower density state. So the density will, this perturbed density will eventually relax back. There will be a time scale associated with the relaxation. And the statement is that if you put in a perturbation of wavelength lambda, and there is a relaxation time for this system, which I call tau lambda, relaxation to thermodynamic equilibrium. If the relaxation time goes to 0, goes to infinity, as the wavelength of the perturbation goes to infinity, such a mode is a hydrodynamic mode. So you see the connection to thermodynamics what one is getting. In the limit lambda goes to infinity, you really have a uniform system. Thermodynamics, there's no space-time, right? So you say pressure, temperature, but pressure where, temperature where? It's the same everywhere in the system. So it's uniform. So a mode for which if you put in a perturbation whose wavelength goes to infinity, the corresponding relaxation time to thermodynamic equilibrium also goes to infinity. Such a mode is called a hydrodynamic. So the examples being densities of conserved variables. So if you have a mass, total mass of the system, which is a constant, the density of the mass, the material density, is a hydrodynamic mode. We also learned from school that the total momentum of a system is conserved as long as there are no external forces. This is something that Newton told us, right? So therefore, if you have a density of the momentum, that's a hydrodynamic mode. If you are close to some phase transitions, order parameters, I'll qualify that in pictures. It's basically the orientational degrees of freedom that we talked about in the first class. By the way, we had idlis for breakfast. How many of you tried to do the discotic phases? Nobody tried? Yeah. What's the point of making idlis? No, I showed a picture of idlis. Yeah. So there are order parameters, orientational degrees of freedom, and such things also become slow modes. Slow modes are hydrodynamic modes. Okay? So this is a general theory which is known in physics, how to write down unified hydrodynamics for crystals, liquid crystals, normal fluids, superfluids, and many other things. So what one is trying to do is to write down a description for systems which are away from thermodynamic equilibrium, close to equilibrium, but away from them, perturbed, and try to see if you can come up with nice equations for this and hope that these will work for living matter too. So examples. For example, the equation that describes the flow of a fluid, I'll derive this equation to give you an illustration, and the resulting hydrodynamic mode is a sound mode. The fact that I'm talking to you and you can hear me is because of a hydrodynamic mode. In a bacterial context, hydrodynamic theories have been used to describe suspensions of swimming bacteria, the cytoskeleton itself. I'll work out some simple models for that. Much larger scale flocking of birds, swarms of fish. So these are basically theories where you take in a description at a thermodynamic level, you extend it to non-equilibrium steady states, look at slow variations in space and time of the relevant degrees of freedom, and write down the equation. Active hydrodynamical theory of the cortex is one. So I just want to emphasize this. So this is really not looking at very molecular detail. So you're not catching hold of molecules. So if you have a glass of water, you can look at it at several levels. You can really think of this as a dielectric medium where you usually resolve the charges on the atoms and so on. Or you can really think of it like a hydrophobic medium, or go up and it's a really colloidal particle sitting in some fluid. Much higher, it's a viscous medium. This is a flow. And much higher, you even remove all that and you just say there is a lattice in which there are water molecules sitting there. So what level should I do? I mean, which level of description should I use? Well, there's no uniform answer for that. It depends on what can you measure. I mean, if you can measure things at this level, it's pointless to write down a theory at that level. If all you can measure is at this level, there's no point to write down a theory at this level. So one would really like to write down a sequence of theories, if possible, which is appropriate to the level of description for which you have experimental data. So it's pointless to think that I'll get a theorist and he'll write down a theory for something for which I have no experimental. It doesn't work. The wavelength of the perturbation. Two molecules, huh? Yes. No. I really want a perturbation. Okay, let's say you push the current, so increase it here. It's actually, this is a finite size system. So it's not a lambda equal to infinity, lambda equal to half the box size. You really would like to look at an extended system, not a finite size system. Boundary effects are important, finite size effects are important. I won't be able to treat them here. So how does one construct a theory? So what one does is just like we had a description for the concentration field by saying that I'll look at a tiny volume, count the number of molecules there, I'll call it the concentration field, and then I'll write down an equation for the concentration field. So we had written down equations for the diffusion of molecules. So intuitively we had a picture that there is a glass of water and I have a drop of ink that I have put there. And what I'm really looking at is the concentration of the ink molecules, not the water molecules. The water molecules is the thing that's coming and hitting the ink molecules. And I'm looking at the concentration field of the ink, and so I'm writing down a diffusion equation for that. But then, if I actually slosh the fluid, the water molecules will also move around. This flow that can happen. So what am I talking about? Is the flow that one is trying to answer the flow of the individual molecules or the flow of some much more coarse-grained description? I mean, individually the molecules will probably be doing random trajectories. It will be really chaotic, really messy trajectories. But one is trying to look at is a much more coarse-grained picture. You maybe take 100,000 molecules, construct some micrometer cubed volume, and look at the volume, look at the velocity of the concentration, look at the velocity of that volume, or the concentration of ink molecules in that volume. It's really a coarse-grained picture. So, but there's no general prescription for how big should the volume be, or how small can you go and still have a hydrodynamic theory. I mean, it's really a rule of thumb. There isn't a specified thing that one can say about this. But how do you get the equations? I mean, this is just saying things in words, but how do you get equations? How do you make quantitative predictions? So the idea, first thing, is to identify the hydrodynamic modes. You identify the conserved quantities, and so therefore the densities of the conserved quantities are the hydrodynamic modes. In a simplistic description, you will write down first order equations and include all terms that are permitted by the symmetries of the problem for the hydrodynamic modes. So I'll show you more examples of these things as we go along. And if you want, you add in noise terms, which are really representing the fast degrees of freedom, the degrees of freedom that relaxed very, very quickly. If you are at equilibrium dynamics, that is, you are looking at a system which you have perturbed from equilibrium and the system is going back to equilibrium, which means the dynamics is still governed by a detailed balance condition. You are supposed to respect the time reversal symmetry of the system. So the time reversal symmetry buried in Newton's laws, in a statistical sense, this is the remnant of the time reversal symmetry that comes up here. So if it's equilibrium dynamics, you must have terms that satisfy time reversal symmetry. If it's a non-equilibrium steady state, that is, you're constantly fluxing the system out of equilibrium by pumping in energy, you do not need to respect time reversals. So that's the general prescription. So let's do a simple example. So let's take a simple fluid at isothermal conditions. That means I'm not going to worry about the energy of the system. So what are the conserved quantities? I know the total mass is conserved and the total momentum is conserved. And so what one identifies is the local density of mass, which is the mass density, and local density of momentum, which is a vector field. And one asks, how do I write down equations for this? Well, I know the total amount of material is conserved for rho. So which means that if I look at the whole system or any tiny volume, so let's say I have a big system and I look at a tiny volume here, and I ask how much mass is there in this system. So the total amount of mass in this system is basically the integral of the density of this volume. Agree? Then you ask, how does this mass change? Well, you did not create molecules or destroy molecules. So the only way the total amount of stuff here changed if something came in or something went out. So that sort of gives you an equation for the density field, which is essentially like that. So the rate of change of the mass, the total mass, will be the integral of the momentum over the surface. So the momentum is basically measuring how much stuff went out or how much stuff came in. So there is a mass density, and the current of the mass density is the momentum density. Yeah. So if you look at individual molecules, they're really maybe doing random walks or doing some chaotic dynamics if it's really looking at individual molecules. So if you catch hold of one, it really looks like a random trajectory. But on this large scale, if the fluid is moving as a whole in one direction, there is a net average motion of all the molecules. Doesn't mean one molecule went this way. One molecule may have done this, come down. On the average, if you wait long times, the whole volume or the whole set of molecules on the average would have moved in the direction of the hydrodynamic flow. If there is a net flow, yes. So the current of the mass density is the momentum. So the time rate of change of the total mass is the flux of the momentum going in or out of the system. The next quantity is the momentum. So you would like to ask if there is a similar equation. Well, it should be a similar equation, right? The momentum is also conserved. So therefore, the time rate of change of the total momentum in the system should be the integral of the momentum current. This is the mass and the mass current is the momentum. So this is the momentum and therefore there must be a momentum current. And there is a mass and the momentum current was a vector. This is a momentum which is itself a vector, and so therefore its current should be a tensor. And that tensor is called the stress tensor. Many people use stress for different things, and this is enough at this level. It's basically a current of momentum. So how do you calculate the stress tensor? Well, you again go back to simple Newtonian physics. So at the end of the day, this equation that we had here will really turn out to be something very close to Newtonian laws of physics. I will not worry about this derivative for technical reasons. For people who know that, it's fine. It doesn't matter for this. I'm anyway going to knock this out. The total rate of change of momentum is the total force. That's Newton's second law, F equal to ma. Now you said the total force on this volume has parts that come from other volumes surrounding the volume that you're looking at, and maybe some external forces. For example, there's gravity, or maybe there is some electric field, and so on. So you'd like to understand this stress tensor that I said, the total stress tensor, which is the thing that appears in the surface integral for the momentum equation, will have the following parts. This is something intuitively obvious to all of us. If you have a tube of water and you put a high pressure here and a low pressure here, you're going to drive flow. Pressure gradients drive flows. So that's one part to the stress. There's another part. If there are different layers of water and the different layers of water are moving at different velocities, you all know the following, that there's going to be viscous drag forces. One layer is moving faster compared to the other, so this guy drags that guy pulls this, and so therefore, depending on the way in which the velocity changes with position, you will get additional viscous drag forces between different layers of the fluid. That's the part that comes from the viscosity. So you take this form of the stress tensor, pressure plus viscous stress gradients, put it into the equation for Newton's laws, and out pops what's called the Navier-Stokes equation for flows. So the Navier-Stokes equation says the total rate of change of momentum is balanced by the pressure gradients, the viscous forces, and any external forces like gravity that you have in this. Yeah? That's the case when you're looking at phenomena like turbulence or weather, where inertia is very, very important. So this is again some example that one does in a high school level physics class. You have a harmonic oscillator, there is a spring force, and you say, after you solve the harmonic oscillator, you say there is a damping force, maybe you put it in a viscous fluid, and so there is a damping force and there is a spring-like force. And so when you try to solve this oscillator, there's a balance between the spring force and the damping force. And you identify regimes where the system oscillates many times or the system just dies down exponentially with time. So the regime where it oscillates several times and eventually dies down at a long time is called the underdamped regime. Inertia is the thing that's driving the oscillations. And the other regime where inertia is so negligible compared to the damping, where the oscillator just goes down, doesn't do any oscillations, is called the overdamped regime. So basically trying to compare, is the inertia of the particle important or is the viscous forces important? If the viscous forces are important, you will get an exponential decay. If the inertia is important, you will satisfy Newton's laws that if you kick a particle, it will continue to move in a straight line unless disturbed by external forces. So biology operates in a regime where inertial forces are almost negligible at the cell and developmental level. At the organismal level, for swimming of whales and all, inertia is important, or your own swimming, inertia is important. At the cell and developmental level, inertia is negligible. So a quantitative measure of this inertia is one thing called the Reynolds number. Let me complete this. So this really measures the ratio of inertial forces to viscous forces. And so if the viscous forces are much, much larger compared to the inertial forces, the Reynolds number is going to be very, very small. In fact, for cellular and developmental flows, Reynolds number is like 10 to the minus 3 to 10 to the minus 6. So inertial forces are almost always negligible compared to viscous. Yes. There you're pressing the system by putting in some pressure. I don't know the exact system. If you're looking at self-migration in some nice biological environment, inertia is almost always negligible. So you could take a cell which really lives in some nice low Reynolds number environment and subject it to flows which are much, much larger compared to what it's never experienced. Then of course it will behave like a particle which is undergoing some large inertial forces. In a natural environment, you can make an estimate and inertia is negligible in physiological conditions. So what does that mean? So we look at Navier-Stokes equation. We said that the Reynolds numbers is very, very small, which means inertia is very, very small. There's only going to be a balance between the stresses, the forces that come from the stresses, and maybe some external forces in the system. So basically this term is negligible. So which means that if you go back and look at in terms of the Navier-Stokes equation, an equation for the momentum density really became an equation for force balance. You balance all the forces at every instant of time. So that's how one constructs a hydrodynamic field, hydrodynamic description. So for a simple fluid like we had now, so we had a density field and we had a velocity field, and we constructed equations for both the density field and the velocity field. Okay? But that's not the whole story, right? Like we discussed in the first class, there are more complex fluids. There are fluids where the microscopic constituents are not point particles or spheres, but really are rods. The rods could be nematic, the rods could be polar. And so therefore, now you need to include the hydrodynamic descriptions of such orientational degrees as well. Particular example, actin cortex is made up of actin filaments, which are polar rods. And you are really looking for the density of actin, density of myosin, the flow of actomyosin, but also maybe the orientation of actin filaments. So one needs to include that part as well. So there are hydrodynamic theories of polar and nematic liquid crystals which have been worked out. One more thing which we have been working using the master equation, chemical reactions. You could have many species of such molecules, A, B, C, and so on. A makes B, B makes C, D, whatever, all possible chemical reactions. You can't change the mass conservation law. You basically convert this to that and that to this and make different amounts of different chemicals. So therefore, you could have chemical reactions too. And so one will have to include the chemical reactions, and so the relative amounts of A, B, C, D, whatever E, and satisfy conservation laws for momentum, for mass, momentum, equations of motion for the orientational degrees of freedom, and the chemistry. So these things have been worked out in the past for physical systems. Now we come to the things that will really be relevant to our physics. So we want to look at active materials. For example, the atomizing cortex. So it turns out that this is a particular class of systems which goes under the name of active matter. You'll hear many lectures about this. I've listed a few review papers, not all of them. The literature is extensive, so I only listed the review papers, not the original papers. You will hear more about this from Frank Ulischer, from Guillaume Salbreux, and others. So a particular example of active matter is the atomizing cortex itself. So there's a conceptual difference between a typical non-equilibrium system and an active matter system or an active system. So the reason is the following. A typical non-equilibrium system, for example, a glass of water, you would heat it from the boundaries. You would heat it at the boundaries, and then the energy would trickle in slowly into the bulk, and then maybe if you leave it there, it will settle down into some thermodynamic equilibrium. Or if you keep continuously heating it, you could get patterns, Rayleigh-Bénard convection patterns that Fernando showed, for example. Active matter is a slightly different analog. Active matter is described as a set of non-equilibrium system where the driving and dissipation of energy occurs at the individual units, not from the boundaries. Okay? So active matter is a novel class of non-equilibrium systems. where the driving and dissipation of energy occurs at the level of individual units. Think of the atomizing cortex. So you have ATP, which is buried everywhere in the cell in a large amount, and the ATP acts on the motors everywhere in the atomizing cortex simultaneously. It's not that the motors which are close to the boundary are the only ones that will get in the energy and then it will trickle down. It's really looking at systems where the energy supply or the energy throughput, that's the proper word, is available at every place in the system uniformly. So if you look at the non-equilibrium hydrodynamics of such systems, additional terms have to be put in. One, for the fact that the system is out of equilibrium, so therefore time reversal symmetry is not to be respected. Two, because of these active things, certain other terms which are not allowed by equilibrium do appear. Particular example, the atomizing cortex. So in a very simplified description, you could do the following. Look at the atomizing cortex, which is really a thin film of actin and myosin motors beneath the plasma membrane of every cell. And as we learned, it's the driving force for mechanical force generation, motility, cell division, polarity, even tissue level deformations, and so on. So in a hydrodynamic description, large scale, long length scale description, one writes down a stress for the meshwork and identifies that there are two possible stresses. One is a usual passive stress. Suppose there was no ATP. So that would mean the system has a bunch of filaments and a bunch of motors, and the motors and filaments are interacting with each other like an equilibrium system. So there could be the usual viscous stresses, pressure stresses, whatever stress is possible, or some anisotropic stresses because of the orientational degrees of freedom. But the system is not out of equilibrium because you don't have energy. You don't have energy flux via ATP. If you supply ATP, there are additional stresses that come up. So the intuitive way to think about it is that there are motors walking on the filaments and the filaments get tugged. And so therefore, on a larger scale description, this essentially enters as new terms in the stress. Those are called active stresses. So the active stresses are a function of the chemical concentration of motors, ATP, and also possibly the orientational degrees of freedom of the filaments. So that's the picture that one would like to build. And I want to particularly work out one example where such a thing comes up. Okay, so this is what I showed you last time. So one thinks of the atomizing cortex as basically a meshwork that turns over very, very quickly on time scales long compared to the turnover time scale on which this turnover happens. The atomizing cortex really looks like a fluid. So one has viscoelastic descriptions, one has viscous descriptions, or even elastic descriptions at the level of the atomizing cortex or even at the level of the tissues. Some of that will be discussed in the main school. So my plan is to give you a set of ideas and a simple toy model which will illustrate the basic ideas of how patterns that arise in active materials are different from patterns that arise, say for example, in conventional non-equilibrium systems like the Turing patterns or the Rayleigh-Bénard convection cells. So I said the atomizing cortex is a thin film of actin and myosin filaments, and it turns over on time scales. And on time scales much longer than the turnover time scales, it really behaves like a simple viscous fluid, though it's an active fluid. Activity really comes from the flows. Can we have the lights off? Turnover is basically the following thing. So there are filaments here sitting at the edge of the cell. They are continuously polymerizing, they are continuously depolymerizing. So think of a piece of rubber in which you have all these filamentous structures, and the structures are locked into each other and you have a rigid network sitting there. Now look at the case where the polymers that's forming the rubber structure are continuously polymerizing and depolymerizing. So any stresses that you would have will relax on a time scale on which these structures are broken. So therefore, the material will flow on the time scales on which this relaxation happens. Definite example, if you really look at the atomizing cortex in, for example, the silicon zygote, This is a picture I showed you yesterday. You see a large scale flow of myosin motors happening there. So like I said, you can actually measure this flow experimentally, and you find that there is a correspondence with the myosin profile that comes up there. And one can actually look at the picture of the flows. So the intuitive idea, like I described yesterday, is that there's a patch of the active myosin cortex with some motors which has a contractile forces. A higher density patch has a higher contractile forces. And so if there's a gradient, you get a flow. So let's make that more quantitative. So what you have is a hydrodynamic description, and I will. So you're really thinking about the cortex, which is some small thick meshwork of filaments sitting beneath the plasma membrane, and you're really looking at a tiny patch and then asking what are the stresses in the patch. You can make it in higher dimensions. One dimension is enough to illustrate the basic ideas. So there'll be two parts to the story. So the stress will have a part that comes from the velocity gradients. That's the usual viscous stresses that we said even for a simple fluid. Now, because the material is active, you have an active stress here, or active contractility, myosin contractility. So the velocity of the actomyosin cortex, a flow. So you can basically look at the actomyosin cortex. You have tagged, for example, myosin molecules or actin molecules, and you see that over a longer period of time, there is a net flow from the right to the left. Catch hold of one of the spots and follow it in time. You can quantify that by doing something called a particle imaging velocimetry, which is just a cross-correlation technique used in fluid mechanics, and you can make the flows more quantitative. And so there is a net profile of the myosin, there is a net profile of the flow. Actin and myosin. So myosin is tiny motors. At this level of description, yes, but it's not a point particle. Meshwork of actin filaments and myosin motors and ATP. And for people who know the cortex, and cross-linkers and severing proteins and many proteins. So the density of the whole cortex is assumed to be constant. But what I'm looking for is the density of stuff buried in the cortex, myosin motors, for instance. It's really like there is a material which is like a fluid, and I have put in some stuff that can diffuse in the fluid, myosin motors, and that's the equation that I'm going to look. The flow is a hydrodynamic flow of the entire stuff. Buried material plus whatever the background material that there is. There are two densities in the problem, and I've assumed that one of the densities is fixed. You can write down a system where you have a total density of stuff, and you have relative densities of the actin, the fluid, of the myosin motors, and so you can work out the relative equations. Now look at the case where the total density is fixed and there's only one relative density which is really moving around, or chemical concentration of the myosin motor. Yeah, simplest description. You can write down separate densities for both of them, it'll get more complicated. Yeah. Which assumption? Yeah. Yeah. You can get a system where you can make some analytical predictions and you can check it with experiments. I'll show you that this can make a prediction about this. I mean, that's why we sort of have a faith in this kind of a description. Right. So in the hydrodynamic description, one says that there is a stress, the active stress or tension in the two-dimensional case. The total tension or the total two-dimensional stress is a viscous part, velocity gradients, and an active stress. And like we said, flows are low Reynolds numbers. So it's basically a force balance equation. And so the gradients of the stress must balance all external forces that are there in the system. Agree? If you go back 2 slides. In a very simple description, one says that, remember, there's a cytoplasm here and there is a membrane sitting here. So you're really looking at the motion of a thin film on a substrate either the cytoplasm or the membrane. And so to a very low approximation, you can really say the external force is essentially a friction that is proportional to the local velocity of the fluid. It's really like a drag force on the fluid sitting on top of a substrate. So this says, therefore, that the force balance equation translates to something like that. So now you use this so-called constitutive equation and this force balance equation, and so you land up with an equation for the flow. Actomyosin meshwork. The lab frame or the surface of the cell or the cytoplasm, some lab frame. Make sense? Okay, so now the ingredient is, we said earlier, the active stresses are really functions of the myosin concentration, the orientation fields, and in principle, ATP, of course, ATP, other cross-linkers, so many, whatever, there are so many complicated things that can come up. So in this picture, we experimentally measured the myosin profile, right? So you can measure the myosin profile here, and you have a velocity profile that is measured. So you can just take this as an input. In a simple description, assume that C active is just a function of M, and take M is the myosin density, myosin concentration. In a simple description, you can think of it like a saturating function where you have some constant times M by M plus some other constant. So now I have measured M experimentally, and I have measured V experimentally, and the theory is saying that these two must have a relationship. So I can analytically solve this, mathematically solve this. I take this experimentally given M, put it in here, that tells me a driving force. I can solve for the velocity field analytically. There are unknown parameters like the viscosity, the friction, the strength of the activity, and blah, blah, blah. So if you do some dimension analysis, turns out there are only two free parameters. And I can ask, is there a set of parameters where the experimentally measured velocity field agrees with my analytically calculated velocity field? And it turns out you can do that. So the black lines are the myosin profile that you measured experimentally. The green dots are the velocity profile that you measured experimentally. The green line is a prediction from this theory. So this, remember, this is a worm which is just about to divide and go on to 2, 4, 8 division cycles. So this is a real living thing, and you are able to measure a fluid velocity here. You're able to measure the concentration of some motor proteins which generate mechanochemical stresses. You're able to write down a theory which connects the two, and not just a question of qualitative matching. If you look at the units, the flow velocity is actually in units of microns per minute, so you can actually get the numbers out. So, okay, so this is some historical thing of this paper. Stare at this equation. This actually is negative axis. I have no idea why they did it like that. Of the active stress, within this experimental data, we can't do anything. Even if you just assume that active stress is directly proportional to myosin, you get a solid line. If you assume it's some this saturating form, you get this dashed line. Within the data, we can't figure out. The motivation for this active saturating form is basically to say that you know that as you increase the myosin motors, the active stress does grow up. But then you can't grow up infinitely. I mean, there'll be some spherical constraints. You can't put too many myosin. So it will saturate after some time. All right? Okay, so in the remaining whatever 5 minutes, or if you can give me another 5 minutes, 10 minutes, I'll just work out one model in detail for looking at mechanochemical patterns in the actomyosin cortex. So basically now we have, so I'll let the movie run and then we'll see how this goes. What one is trying to look at is the following. In the previous slide, we gave a profile of myosin experimentally measured. We put it into the flow equation and then predicted the velocity and compared with the experiment and saw that this picture is a good description of this model. We would now like to ask the following. Suppose I have a theory where myosin itself has a dynamical evolution equation. The dynamical evolution equation is the usual diffusion, usual linear first order reaction that we've all beaten to death yesterday. And the new term is advection. So this is basically saying that if you have a column of fluid and you have ink put in that fluid, and if that fluid is moving, it will carry the ink along with it. So that's the advection term. So you have now a closed set of equations, an evolution equation for the myosin density, force balance equation for the velocity field, and the claim is that these two equations, just like the Turing equations that we had, has a set of instabilities that will lead to spontaneous patterns coming up. I want to do that and then stop for the day. So the picture is basically the following. There is a diffusive flux that if you have any concentration fluctuation of M, myosin, the diffusive flux tries to flatten it out. Diffusion always tries to make things flat. On the other hand, there is an advective flux that's coming because myosin generates active stresses generate flow, and the flow is trying to pull in material. It's a contractile forces. So there is something trying to push out, there is something trying to pull in, and so if you can balance them and go into a regime where the pulling in is more than the pushing out, you will start getting accumulation of myosin. And that's essentially where the patterns come. Yeah. It can also unbind. You can bind, unbind, you can do some for tugging, it can unbind, it can move around, then rebind. The unbound concentration does not, it's only the bound concentration that goes in. I'm not going to do those details about that, then it will really complicate. So you'll have to write down a separate equation for the bound myosin, for the unbound myosin, for actin and myosin to be together. All right, I just want to do a linear stability analysis for this theory and show you that just like the picture that we had yesterday, you can start getting atoms. That is enough. So we have an equation for the myosin, which is basically advection and diffusion and a turnover. I will use R here because I want to use K for the wave number. And you have the equation for the flow. So there is obviously one homogeneous steady state. Which is m equal to m naught. So like we did yesterday for the Turing patterns, we would like to ask whether this state is stable. So this will turn out to be much more simpler than the Turing patterns. So we say we put in a perturbation, which is m naught plus delta m. And just like yesterday, we say m naught plus summation k delta mk e to the ikx. Thanks. V equal to 0. So if m equal to m naught, which means m is homogeneous, these derivatives vanish, and so the only solution that's possible for this is v equal to 0. So we would like to understand if there is a non-trivial state possible for the system. So we will do this in the limit when the perturbations are small. So that is delta m by m naught is much less than 1. So we will retain terms only to linear order. So first let's look at the flow equation. You will immediately recognize that if I retain terms to linear order, this is essentially C star times m of x. This does not appear. Times the derivative of this function. Let me rewrite this in the following way. C star times some function of m. That function of m is essentially. If you do a linear stability at the linear level, you'll find that eta k square delta Vk gamma delta Vk minus Ik C star delta m times df by m evaluated at m equal to m star. So I'll call this something, capital F, let's say. So you're basically saying that delta Vk is minus Ik delta F divided by eta k square plus gamma. This is basically saying, if you give me a myosin profile, I know the velocity profile. That's all is the statement about this. So we've assumed that we're looking at the linear level, which means this derivative with respect to m evaluated here and retain only first order terms with respect to delta. So that's the equation for the velocity field in Fourier space. So now we go to the concentration equation. And again, if you follow the same procedure that we did yesterday, you will find delta mk is minus dx, which means basically Ik times m naught. Sorry, this is not m star, this is m naught. M naught is the uniform state. Times delta dk minus dk square delta k minus, what did I call that? R. So now we substitute the delta Vk from here. And then you'll get an equation for the evolution of the perturbation. Will be essentially k square by eta k square plus gamma d star f m naught minus d k square minus r delta. If you remember from last time, this is essentially the same thing as, so this is essentially the eigenvalue that we were looking at. And so if this thing can actually go positive, the homogeneous state is unstable to small perturbation. So the condition for an instability of the homogeneous state is will be positive. So you see the following. If there is no active stress, which means C star is 0, then you go back to the usual equation that we had earlier, pk square minus r. So this can never go negative. And every mode, like we discussed yesterday, if you take a diffusion equation, perturb it, any mode that you put will always go back to a uniform state. So this can become positive only if the C star variable, first of all, is positive, and secondly, can become large enough that it can make the whole thing go positive. Then the growth rate, the kth mode, can actually become positive. So in a sense of a dispersion curve, so what you get is basically the following. So for small C star, you get a curve that looks like this. So C star is small, which means every mode has a negative growth rate, which means every mode will just decay back to the uniform state. As you keep increasing C star, you will hit a point where this will just touch 0. That's the critical C star. And if you increase C star further beyond, I don't have other color chalks, you will get a pattern, you will get the possibility, the pattern basically coming from an instability. That's the instability that you're seeing here. So this length scale of the pattern will be set by a combination of the diffusion coefficient and the turnover rate. And so the number of spots that you would see will be proportional to the ratio of the system size to this length. Okay. I don't want to go into those details. It turns out that this system also has oscillatory patterns possible. If you include 2 species, it can oscillate. We've recently shown that if you have even 1 species, you can get oscillations, but not at the linear stability level, but at a more nonlinear level. I don't want to go into those things. I want you to appreciate the following, that this is a pattern which is essentially a clear mechanochemical pattern. This is the coupling to mechanics. Chemical concentrations generate mechanical stresses. Mechanical stresses drive flows. Flows couple back to the chemical transport equation and change the chemical concentrations. If you did not have this coupling, you would not get patterns. All right. So, but just to emphasize that these are sort of mechanochemical analogs of Turing patterns. So they share the same troubles with Turing patterns. They come from an instability, which means you don't have control on when the pattern comes, where the pattern comes. So the real problems that involve mechanics will couple mechanochemical patterns to signaling. Sources and sinks of flows and blah, blah, blah. And the claim, or the claim or whatever, the work, by the way, I should mention references. Sorry, I've been tardy about mentioning the references even yesterday. So this model was worked out in a paper by, yeah, I have it here. So Boyce et al. In PRL 2010. So therefore, you should really look at mechanics plus signaling, and so therefore you can start getting controlled mechanochemical patterns. And like I was mentioning to you yesterday, the pattern in which this polar pattern comes up on the surface of the C ligand zygote, driven by the same active mechanochemical flows that I was discussing, in addition to certain signals that come from the nuclei, the claim is that this is really a mechanochemical pattern. So one can actually work out a full theory for this. Not this. One can work out a full theory for this. It gets more complicated, more terms, but you should be able to realize the essential ideas. Chemical concentrations, advection, diffusion, complicated biochemistry. Another chemical, advection, diffusion, chemistry. The mechanics part, the motor molecule, advection, diffusion, complicated chemistry. Finally, the flow part, the mechanics part of the story, which generates flows. So all these things get coupled. So in addition, there are signals that come from certain other extraneous factors, the nuclei in this case. So one really has control here in the sense that the signals tell you when to start the pattern, where to start the pattern. The mechanics and the biochemistry conspire to create this polar pattern. M and the A and P, there's one term here. I mean, okay, that goes into the chemistry. There isn't much of a interaction. This is one of the things that we figured out at a minimal level. More details about the chemistry I can tell you later. There isn't a direct chemical interaction at this level. The driving is primarily because the M generates velocities. The same velocity appears in all three equations. There is that coupling. There isn't a direct chemical coupling. At this level of description, we found it's not needed. Except there is something driven by A, and there is some external signals that come in. Okay, so it's just like we had the case where we were looking at a given myosin velocity profile and trying to see if the given myosin profile can be used to predict the velocity field. So it turns out in this case you can take it further and you can actually predict the spatio-temporal evolution of the two polarity fields, myosin motor field and the velocity field, and all of them match in space, in time. Okay, sorry, I've taken too much time. Stop now. Yeah. And mechanics. All right, so I'll stop there. Let's take a coffee break and come back for the yeah. Can people, who wants to go for coffee break, can go and finish your coffee and come back. Yeah.